went to Portville Central Senior High School. She is a Phi Eta Sigma Honor Society member, a Tri Beta Biological Honor Society member, the current treasurer of Chemistry and Biochemistry Club, and is on the Mercyhurst cheer team. Treasurer and stunt.
My voice pretty quiet.
Oh, okay. She's on the cheer team. And after graduation, she hopes to find a job within biological and chemical field. So I'll let Tori take over with our last presentation on MDH, but I promise it's going to be a little bit different.
Yes. Yes, thank you for the introduction. Hi, I'm Tori Ann Morrow. This is my presentation on phospho-mimic mutations of malate dehydrogenase, investigating the impact of post-translational modifications on enzyme activity and metabolon formation. That's a mouthful. Yes. But like Dr. Peretti said, it will be a little different from the other presentations we've seen today. So I want to give a little bit of a background on metabolism. We've heard it before, but just to reiterate, metabolism is the process of converting fuel into energy, specifically ATP, or like we like to call it, our money. And the specific part of metabolism that we are focusing on is the citric acid cycle, which is occurring after glycolysis, and that's when a pyruvate is converted to acetyl-CoA. And then we're gonna get all around this little cycle and get to our area right here, which is when we get to our enzyme malate dehydrogenase, which converts oxaloacetate into malates in a reversible redox reaction. It's also multifunctional. And we move around and do all of that with metabolons, which are complexes that are formed between enzymes of a metabolic pathway, and the metabolons are formed with protein-protein interactions. And this image right here, we've seen it before in our previous presentations about metabolism, but just to reiterate that this is not all happening at once. It's more of a possibilities that could be happening. And with those possibilities, we're going to be talking also about post-translational modifications, or PTMs, which is when we modify the amino acid side chain of a protein by adding or removing a specific group. And the specific PTM that our research focuses on is phosphorylation, which is the process of adding a phosphate group to a molecule. And you can just see here that there's a lot of different kinds, but we just focus on phosphorylation. And then with that, with those PTMs, we also get phosphomimics. Phosphomimics are amino acid substitutions that mimic a phosphorylated protein. And so for an example, you can just see here that we have serine, and here's a phosphorylated serine. And then down here, we also see this aspartic acid. Now, that aspartic acid mimics that phosphorylated serine because the charges are both negative and the sizes are similar for the structures. Now getting into more of my focus for the presentation, I focus a lot with the structure of malate dehydrogenase. So this lovely thing is just a bunch of sequences of different malate dehydrogenases. We have prokaryotes, single-celled eukaryotes, a human, and also plants. But the big picture here is that MDH itself is containing of about 317 amino acid residues, and of only about 15% of that is identical, which I'm going to get into those specific identical pieces a little bit later. But what's primarily important about this is that you can see that while the sequences here, they don't look the exact same. But you can look at the top and you can see that they're all making the same types of structures in the same spots, which these arrows represent beta strands, and these spirals represent alpha helices, and they can also be represented by the corresponding colors, the orange for the beta strands and the magenta for the alpha helices. So since that the sequences aren't the exact same, but they're creating the same structures here, you can conclude that nature is going to preserve the structure of an enzyme, and therefore it's going to preserve the function instead of preserving the sequence. So my focus is how we visualize the structure. So I want to give a little bit of history about visualization. Back in the 1950s, John Kendrew found the structure of myoglobin in sperm whale using X-ray crystallography, and doing that he was able to figure out a coordinate system. Now, to understand how the coordinates would work for structure, here's an example. If I were to take all of you and try to recreate everything in this room in a different lecture hall, I would need to give each and every one of you a set of coordinates so you would know where to go exactly and how to stand and all that. Kendrew was able to do this exactly when it came to building his structure. But this is the 1950s. We didn't have computers like we do now, so how was he supposed to build that structure? Well, he built it right in his room, his lab. And I think that's really cool because he was able to immerse himself and physically be in the structure of that myoglobin. And it's really similar to what we do today with structure and those coordinates. We still use coordinates when it comes to figuring out structures. As we're doing research now, we have to upload any new coordinates for our structures into the Protein Data Bank, or PDB. This is a repository of information where we can figure out what proteins can look like, how they're found. We can also find these things called accession numbers, which is a 4-character code that is pretty much an identification number and tells us a little just how we can find and identify that structure. So just in this image right here, this is 2-DFD, which is human MDH, and that's going to be primarily what we focus on since we're working with our human malate dehydrogenase. But I want to talk about this picture just a little bit. This picture is from PDB, and it is a 3D image, which is great. That's what we want to be able to see, right? The only downside is that on this screen, it's a 2-dimensional space instead of a 3-dimensional. And we'll see that, how we can fix that in just a minute. But before we can get into knowing more about malate dehydrogenase's structure, we need to learn about structure itself and how that works. So I'm going to give a little demonstration here. So we have my little thing. Our primary structure, which is just the amino acid sequences. And then that's going to go and it's going to bend and change its shape and come together and making those beta strands and those alpha helices. It's changing. And that's our secondary structure, like you can see here. And then those secondary structures are going to start folding on itself, coming together. It's going to come together, and then we have what is known as tertiary structure, and that's going to be a monomer. And then when a bunch of those monomers and tertiary structures come together, they're going to form quaternary structure. And the quaternary structure for MDH is a dimer, and that's how MDH, at least for human MDH, works best. So we're going to next talk about building MDH structure. Now, this is an image of MDH in E. coli, and I really like this image because it shows really well the different pieces of the monomer before we get into the active site. Now, this area right here, the gray, is the nucleotide binding domain, and that is where we're going to find our NAD. Now, the blue area down here is the catalytic substrate binding domain, and that's where we're going to find our oxaloacetate or our malate, depending on the chemistry. Now we're getting into the hot and heavy, the active site of malate dehydrogenase. In the center here, you're going to find malate calling all the shots, and there are 6 structures all around it, but only one of those actually does our redox chemistry, and that is the NAD over here. We need to have that NAD to do that redox chemistry, but it's gonna spoil some things. This right here means that the structure is actually a lot bigger, but the picture focuses on this area because that's our B vitamin, and that's the part that we really need for that reaction to occur. It takes away this hydride, and then over here we have this histidine, and that's going to be able to deprotonate the malates, and that is able to do that work because this aspartic acid right here makes the histidine a better base. Now you're probably wondering, well, what about these three? What are they doing? Well, those arginine residues, they are there to help preserve the position of malate because they have positive charges and the malate has negative charges. So back to this, you probably noticed that there were little numbers next to all of those structures around malate, and those are showing where they would be located in our primary structure. So these little asterisks that I've highlighted here, they show, firstly, these ones are for the arginine residues, and you can kind of see that they're not very close together, but when we just saw the image of the active site, they were pretty close. And then you can also see something similar with the histidine and the aspartic acid right there. Again, in our last image, they were pretty close together, but in primary structure, it's not. And that's just a really good example of just showing how primary structure is not amazing when it comes to actually seeing your structure and actually understanding where everything is. So like I said before, we visualize in a 2D space, on screens, on paper, and that's how we've done it for such a long time. But what I really want to make sure that we're able to do is be like Kendrew and immerse ourselves into our protein. So in the lab, I've been doing that with virtual reality. With virtual reality, we're able to see everything in more of a 3D space. And the program we've been doing this with is called Nano. It's really nice for visualizing the structure, for actually being able to immerse yourself in the inside of your protein. And then you're able to highlight specific areas of the protein itself and able to see where our potential mutations would be. Now, this is a video of what I've done in the lab specifically. And I just want to show you guys it because it's really cool. Okay, so now here you can see me uploading the PDB ascension number to DFD. And here we are. This is our full structure. It's really big, but we can actually change that. We've made it small, and we can practically hold it in our hands. And I'm going to zoom us in and pretty much pull us into that active site. And then you can see that we've gotten to the full structure of NAD. And right here you can see the 4-carbon structure of malate. And right around it, all of those other conserved structures we talked about, like this is the histidine, and how that can deprotonate malate. And the other regions are held in position with the arginine residues. There's one, and here's the other. And this is our last arginine residue. And what's also really great about looking at the structure with these is just being able to just see it. I love being able to see it. And there's that aspartic acid again. It's adjacent to the histidine right there. And that's going to make it, again, a better base due to the charges and the catalytic reactions there. Just showing off a little bit here. just what exactly we're looking at with that NAD structure. It's really cool. And now I'm just going to kind of zoom us out, pull us out, so we can see about how small the active site really is compared to the entire structure. And what's great about Nano also is that you can orient the structure itself into any position you kind of want. Like, this way we can look at the dimers really well. You can also see that there's spirals and those arrows again, like we saw with that image with our very a lot differences of structures. And here is where I'm showing highlighting and how you can highlight as well. And now we're just kind of walking around and going through, trying to see our structure some more. But the spirals are the alpha helices and the arrows are the beta strands. So those are just our primary structures, again being shown, our backbone. So there's not enough time to really go through everything in the labs, but I did help with some of the experimental work that we've done as well. Again, my work today was more about structure, but if you were here earlier, Michaela talked about her work with overexpression, and the other day Maria talked about her work with protein purification and characterization, and Danny also talked about her work with seeing enzyme activity using enzyme assays. And speaking of all of that, what we're going to eventually be able to do with this is see our different mutations and where they would be. This is just a structure of MDH in a program called PyMol, which is just another visualization program. And my specific mutant is S45D. It's located right over here in yellow. We didn't get to do much work with it specifically, not enough time, but in the future we're going to be able to do more work with that as well. So using virtual reality, we can see and try to edit malodehydrogenase, and we can use this to help us with our phosphorylation, seeing how that affects enzyme activity. So for the future, I mentioned how we're going to be able to use this process with seeing our mutants in the future. So one thing that we kind of noticed when I'm going through working with Nano and PyMol, it was kind of hard to find our specific, where our specific mutants would be, and also trying to edit them was something that we weren't able to really figure out that much, which kind of bummed me out a little bit. I really wanted to see how that would work. But we talked with Dr. Provost, who is Dr. Peroni's collaborator, and he told us about this program called AlphaFold, which is an AI program that allows us to actually edit where our mutations would be and see those phosphomimics. And then with the AI, it will fold that new sequence into how it's supposed to be and give us new coordinates. And then we can use those new coordinates and put them into programs like PyMol and Nano to be able to visualize the new structure well. And so I really just wanted to thank Dr. Peroni for all the help that she's given me with this and letting me play with VR. That was a lot of fun. I'd like to thank everyone in the lab group for all the help and for just being really great and just everything, and also the department for allowing me to have this opportunity. I enjoyed my time a lot, and I'm going to miss this. Thank you.